Hi everyone, it's Mr. Sinti, and thank you for joining me in this ephemeral historical look at CRISPR-Cas9 gene editing. That's right, this video is going to take a look back historically at some of the crucial experiments, and I would say also a look back at various scientists involved in those crucial experiments, not so much for content knowledge, which is important, but rather for inspiration. I find as a researcher, I enjoy looking at some of the great discoveries of the past, not only because of their contribution, but for their for their modeling of insightful creative thinking. I'm always marveled by, the more I spend time in science, I'm inspired by creative, imaginative solutions to problems that we have, and it's almost on equal status with understanding in terms of like mechanics of how things are operating. And so this this look back at CRISPR-Cas9, I'm going to begin the conversation back in the late 1980s, around 1987 in particular, where Japanese scientists were working with E. coli. No surprise there, and how important E. coli has been to our knowledge of molecular biology over the years, and still continues to contribute remarkably. Um, and nevertheless, they were working with a pretty common enzyme, alkaline phosphatase, and they were sequencing certain regions of DNA, and they were noticing, I would say, unusual uh, repeating segments that were interrupted by spacer DNA, like interspersed in clusters. And so it was odd discovery. And at the time, not much was made of it other than noting it in the literature, which was really important as it turns out. But as we'll see, it contributed to where we are with CRISPR currently today. And so CRISPR, as you may be aware, is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. So that is a mouthful, and thus the acronym. And we'll try to unpack that in this particular video, looking at the historical perspective of CRISPR. And so there's a picture of scanning electron micrograph of E. coli there. And so um, CRISPR-Cas9 in general is an adaptive immune response system that protects prokaryotes and archaea from bacteriophages. And so bacteriophages, as shown in this image right over here, are viruses that can, if you will, let me see if I can draw this here. So these viruses can attach onto E. coli, and what they do is um, sort of viciously, they inject their nucleic acid into bacteria, and as a consequence of them injecting their viral DNA into E. coli, they commandeer the host cell, which is the E. coli, and they use the host cell's equipment, meaning ribosomes, to replicate the protein. They use uh, the various polymerases, and they turn the E. coli into a viral factory. And so what the bacteria has evolved is a defensive system. And so what they can do is when, let me see if I can then illustrate this a little bit more clearly. Say this is the viral DNA going into E. coli, and I'll just label that viral DNA. What the bacteria have been able to do is develop a mechanism, and more about this as the video unfolds, but just simple, developed a mechanism that is different than restriction endonucleases, which are, if you're familiar with those, a class of enzymes that are capable of recognizing certain sequences, specific restriction sites, and cleaving or cutting DNA. This is more of a targeted response and a more adaptive response, and so it's new. And how it works briefly is that these cutting enzymes, endonucleases, are guided to this particular viral DNA sequence by an RNA sequence that will lead them right to this particular viral sequence. And this enzyme will then cut the DNA, and therefore the bacteria is protected. And it's like, well, that's you might be thinking that's useful and quite remarkable. Like, how is it that the bacteria possesses RNA that would cleverly match that of a viral DNA? And so, as you may imagine, sort of like how our own immune system works briefly, is that once exposed to a particular germ, or in this case, a phage, the descendants of the infection will develop an adaptive, meaning responsive immune system to that particular phage. And how they do that is that they incorporate a portion of the virus DNA into their own chromosome. Okay. And so, though this video isn't about how the mechanism works, I think it is a little bit useful in terms of establishing that concept. And so here's E. coli, the cell, and we'll say that the virus again is injecting its nucleic acid viral DNA into the host cell. What the bacteria is able to do with CRISPR is using an enzyme called Cas9. There are others, but this is one of significance. Cas9 is capable of cutting the viral DNA and therefore protecting itself. And so that's the short version of this conversation in terms of how that's working. And so the use of CRISPR-Cas9 in gene editing was really, jumping from 1987 to 2012. It really, began becoming really popular in the literature, and it was in the mainstream spotlight of molecular biology when major groups at major universities started harnessing this mechanism of that bacteria possess to fight off viral infections, to modify targeted regions of other genomes. And so they're using the protein complex that cuts DNA and developing what will be known as a single guide RNA to target any particular sequence that was felt could be edited and altered or disrupted in some capacity. And so this is where things get really interesting in terms of biotechnology and the potential of this CRISPR-Cas9. And so just to shout out for a couple of these scientists. So there's George Church, is working in CRISPR with woolly mammoths. More about that in another video on applications of CRISPR out of Harvard University. Jennifer Doudna out of the University of California, Emmanuelle Charpentier, working for the Max Planck Institute in Berlin, along with tough to pronounce the name, MIT Broad genetic scientist Wang Zhang. I'm sorry about the mispronunciation of that. And so Jennifer Doudna and Emmanuelle Charpentier also should be noted, won the 2020 Nobel Prize in Chemistry for their involvement in CRISPR-Cas9. There are other scientists that I am confident will be contributing to the discovery, and there will be many more Nobel Prizes coming in this direction. And so what's interesting about this, getting back to the history of this, is that critical insights for CRISPR and how it works and how it all began came from the emerging discipline of, I think of it as emerging, not to be old, the emerging discipline of bioinformatics, which is concerned with acquiring, storing, and analyzing various biological data, like nucleotide sequences, most specifically, but also amino acid sequences. And so comparisons can be run between organisms when you have a genomic database. And so the National Center of Biotechnology information site is a good one. And so, as I was mentioning, in that late 1980s, group of scientists working with E. coli noticed these unusual repetitive DNA sequences. And so just a little view at this. And so repetitive, meaning that this particular sequence shown here in dark occurs, and then it occurs again and occurs again, and it's flanked by or interrupted by these spacer sequences right here that were not repetitive. And so these were unique, and these were repetitive sequences right there. And so the colored regions were referred to as spacers in between the repeating units. And so it was also detected that genes adjacent from these repeat spacer sequences were known as Cas or CRISPR-associated sequences in the genome. And these particular genes are associated with producing the protein that cuts DNA. So this is the sequence that creates the protein that cleaves the DNA sequences. And so you have repeats and spacers in between the repeats. And so these segments had never been seen before. And at the time, looking back, obviously, looking back retrospectively, it may appear to some today that what wasn't it clear to scientists at that time what was going on, but not necessarily, because it's difficult to predict sequence going from sequence to function. We're still not that good at doing that, but with the lack of the DNA sequence data was really the hindering effect. The more DNA is sequenced over the years, we've been able to add to the bioinformatics gene banks of data, and so therefore we're able to run scans and look for comparisons in terms of what organisms have these sequences and what their functions are. And so just to give you a little look at what this might look like when you're sequencing DNA, it usually comes in the form of a chromatogram DNA sequencing like this. And so these pink areas were areas that were repetitive in the E. coli chromosome. And so you can see here these peaks represent the A, T, C, and G, if you're unfamiliar with this. And so there's repeat, spacer, repeat, spacer, repeat. And so what was interesting was that these repeating units were palindromic in nature. And so that palindromes are things that are read and have the same meaning when they're read both forward and backwards and reverse, like never, odd, or even. So the repeats are palindromic. And the sequences were separated by spacers of anywhere from 25, 75 bases that were not palindromic. Okay? So there seemed to have been a pattern. It was unclear, again, the meaning and purpose back in the late 1980s, early 1990s, but the spacer DNA in between the repeats was not identical. And so each segment of the spacer DNA is unique. And so that in and of itself is a puzzlement. The repeats are a puzzlement. The fact that the spacer, each one of them are unique, you got to wonder about it. And so that, again, that I pause here may be obvious, but this is where science begins. It looks at things, it wonders. It's certainly a question that was needing to be answered. But jumping ahead to the 2000s, what researchers found upon sequencing and then again applying bioinformatic analysis is that these spacer DNA sequences match up perfectly with bacteriophage DNA. So let me see if I can illustrate that. I'll go blue here. So here's a bacteriophage. Here's the DNA. So this DNA, when it infected a previous bacteria, the bacteria was able to take a portion of this viral DNA and actually include it in its own genome, its own bacterial genome. And then you're like, well, what's the point of that? Well, if you're following, I didn't really expand too much, but this is what is going to create right here, this particular area, a segment of RNA when it's transcribed that will, in a future infection, will pair up perfectly with the viral DNA And then if this thing is associated with an endonuclease, like for example, like this, it would then cut the foreign DNA and protect it. And so it was becoming clear that what was going down was that um, these sequences are really important. So getting back historically to the year 1993, scientists had chanced upon these palindromic sequences when they were studying other classes of single-celled organisms. I mentioned this before, the Archaea. And so Archaea also prokaryotes, but not necessarily so closely related to bacteria. So they look similar to bacteria, but they're very different in terms of their genetic heritage and also their biochemistry. And so briefly, they're sort of extremophils, meaning that this particular bacteria, Archaea, that was being studied and looked at was a halophil, meaning that it's salt tolerant, that it can survive in these harsh conditions like this. And so they discovered that they also possessed this series of repeat, spacer, repeat, spacer, spacer, repeat. And so when that popped up, researchers wisely noted the fact that, wow, let's take a look in the literature and see if anyone else has discovered this before. And so they looked back and they discovered that the Japanese paper, and they struck gold. And they were like, hey, look at this. Back in 1987, this same phenomenon was present in E. coli. And so they excitedly reasoned that, and wouldn't you, that if these palindromic sequences were present in both archaea and in bacteria, they must serve an important function and purpose. I mean, here's a little phylogenetic tree here showing like evolutionary relationship between bacteria and archaea. And so the conservation of those sequences, even in different domains, was critical for the appreciation for its importance. And so that, I think that's kind of cool. So jumping up to the 2000s, so the discovery of the similarity between the sequencing between archaea and bacteria was important. But then the spacer sequences, again, I'm going to come back to this, were basically aligned perfectly with that of bacteriophages. And so it became common in the scientific communities in the early 2000s that we were looking at some kind of adaptive immune system that bacteria had against viruses. And so the term began to increase in the literature. Predictably, the clustered regularly interspaced short palindromic repeats, or CRISPR. And then early in the 2000, researchers turned their focus from the repeating segments to the spacers themselves, which are the sequences of the viral phage. And so at this time, when you upload those sequences, so you're extracting E. coli, you're lysosing E. coli, you're extracting DNA, and then you're sending it in for sequence, and then you get the sequence and you throw it into the NCBI website, and you hit the blast search. And the hope was to identify, like, is there anything known that we've sequenced that is similar to these spacer sequences? And guess what? This paid off, because the sequences of the spacer, as I mentioned before, this was also a matter of striking gold, aligned perfectly with bacteriophages. And so this was an eye-opening moment, that this was clearly an adaptive immune system that bacteria had, and it wasn't known before to possess. So that in and of itself was pretty interesting. Okay, so that an example of this was that one of the spacers in E. coli matched up perfectly with P1 phage, which is a phage that is known to affect E. coli, as you can see in this picture here. And so they were indeed belonging to phages. And this is a picture of phages attacking various bacteria right there. And so, why would DNA end up in bacteria? Simplifying this for a second, it's because the bacteria is trying to remember previous infections from the virus. And so, if it can take a portion of the virus DNA and incorporate it into its own genome, then it would be able to respond specifically to those infections at a later time. Pretty remarkable, to be honest with you. So perhaps, and then definitely, it's an adaptive immune system, and it was a way in which bacteria can fight their old nemesis, the bacteriophage. And so, pretty cool discovery. And after a short amount of time, many spacers had been sequenced, and most of them matched um, phages. And so, in 2004, correlations are being made further between spacer DNA and bacteriophage DNA. And so what I find another interesting point is the more a bacteria and its descendants, of course, have been infected, see these colored sequences, the better their resistance would be. So if you're thinking creative, like, normally you would think, all right, well, why am I so excited about bacteria defending themselves against against viruses? Well, again, creative thinking would be like, well, we use bacteria for a variety of purposes. And it's like sometimes having bacteria that are resistant to phages could be economically useful. And so I'm just throwing that out there. Spoiler alert. So uh, and I also want to mention out that this is not the first time that bacteria have shown resistance, if you will, to phage. Sometimes there's resistant mutations like receptor proteins, as shown here. The receptor protein can mutate, and therefore that can convey resistance. But this is different. This is the sequences are being placed, viral sequences are being placed in between repetitive sequences. And so these green segments are viral DNA. And so, here's the story. This is, I find, very interesting. And so again, what possible economic use could this be? The fact that bacteria can become increasingly resistant to viral infection. Well, the yogurt industry where also works heavily with bacteria, as you might imagine. It's not just basic research. And so they were looking at the serious problem that is faced every year with a lot of their yogurt culture. Their bacteria get infected with phages, and it reduces their ability to produce yogurt. And so the goal would be to produce a starter culture of bacteria that's better able to fight bacteriophage attacks. And so this is occurring and has occurred. And so the phage infection, the starter culture, is widespread, and it's a significant problem. And so, as it turns out, that I was new to this knowledge, and so I found this to be kind of interesting. I wanted to share it with you. Is that estimates show about 2% of yogurt, also cheese production worldwide, suffers from phage attacks. Didn't realize it was such a big problem, but it is. And so researchers began to notice CRISPR while they were sequencing bacteria for yogurt and cheese production. And they originally they didn't understand the purpose, as I was mentioning, of CRISPR sequences. But then they started to make the correlation between increased resistance to phage when they had increased number of spacers sequences in their genome, and so they were better off to fend. And they also identified in parallel there are genes that are associated with those CRISPR sequences, the repeats and the spacers. And these are the genes, as I was mentioning, that produce the endonuclease Cas9 that is basically a helicase that will unwind DNA, in this case viral DNA, and it will also cut it. And so they're like helicases and nucleases, the Cas9. And so jumping to 2008, it was shown that the CRISPR RNA, once transcribed from the bacterial DNA, is directly responsible for CRISPR-based resistance. And so at this point, it's like, well, let's try to show that more specifically. And so they tried to create an artificial CRISPR array with the basic fundamentals of okay, repeats, spacer sequences, repeat spacer sequences, and they tried to target it against specific genes in lambda phage. And so this is a type of bacteriophage, lambda, if you're familiar with it. And then as predicted, that experiment showed that it was resistant to, the bacteria was resistant to lambda phage. And it was the first case of directly programming a CRISPR-based immunity specific to lambda, and it was shown to be like a flu shot, if you will, a vaccine for bacteria. And so we're starting to make bigger headlines as time goes by. And so bacteria use this system, as I was describing, to defend themselves against viruses, but scientists wanted to harness it. As I was mentioning, the yogurt community, they wanted to harness it and to try to immunize bacteria cultures. And so the dairy developers were sort of the beginnings of this. And so they exposed bacteria to particular viruses and collected those strains that managed to survive the attack. And so the hypothesis there is that upon subsequent infection, they wouldn't die when exposed to all those bacteria. And so, again, that's nothing new, but it's the first time that they were trying to select bacteria for CRISPR immunity as opposed to receptor protein immunity, for example. And so this classic strategy has been employed for a long time, but again, not with CRISPR in mind, and the spacers in particular. And so the yogurt community has been busy culturing bacteria, and they've been isolating these CRISPR cells, and food scientists, if you will, are growing up new cultures repeatedly and then challenging them over and over again with different phages to increase their breadth of encoded resistance. Pretty cool. And so, the trick is to use a diversity of phages. And so you're just constantly exposing your starter cultures to a variety of phages. And every time you do that, their immunity gets better and better and better, and they're able to survive. And so with many thousands of different phages, the immunity is growing. And so you're like, well, I haven't heard of this. Well, it's making the news. Like, for example, the company DuPont in 2007, began vaccinating bacteria strains. And in 2012, the company announced its first commercial application of this, and a new type of pizza cheese that is really resistant to most viruses. And so it's called Choose It Swift 600, and it's doing well. And so, it's very robust, if you will, in terms of the bacteria that converts the milk into cheese. And so it began in an interesting fashion. I mentioned in the late 1987, in 1980s, but in earnest, it was where things started to really progress. And I wanted to just emphasize this is in the year 2011, Emmanuelle, Charpentier, and Jennifer Doudna began collaborating. And after a meeting at a science conference in Puerto Rico, and they went to a cafe, and they were talking about their work, specifically with the Cas9 protein. And this is where, over a cup of coffee, creativity comes in, and it's like, well, what can we do with this? Like, this is what bacteria are doing, but what can we do? What is the potential of this? And so this was a remarkable moment, because after that work, the tracer RNA, which is this segment of RNA. I was mentioning the repeating area. The repeating palindromic sequences sometimes form upon transcription, the conversion of DNA to RNA, form when it's palindromic, it tends to form a hairpin loop of double-stranded RNA. And it's this tracer RNA that scientists believed associated with the Cas9 endonuclease, and it's able to hold on to the protein. So this is where it binds. And the CRISPR RNA sequence in the front right here is how it identifies the particular target sequence for the endonuclease to cut. So this is where it binds to a specific sequence, and this is how it attaches to the protein. And so what was done is, to make matters easier, was these are two separate pieces of RNA in nature in terms of the bacteria producing this. But in the lab, they fused these CRISPR RNA and the tracer RNA together, making something called the single guide RNA. And this was a significant breakthrough, because at this point, this could be introduced into a variety of cells via plasmids or any other form of transformation. And this, all you would need to do is change the targeted sequence of the single guide RNA, and it would locate any sequence in any organism. And so in 2012, this breakthrough meant that researchers not only all you had to do is redesign one component of the single guide RNA, which is to create the molecular scissor to target any sequence. So that becomes very simple. That becomes very cheap. That becomes a very, very powerful gene editing tool that we could use, humans can use, to cut DNA and edit DNA in the future. So this single guide RNA is a big breakthrough. So 2012, scientists combined the CRISPR system with the protein Cas9 and demonstrated how it's a very powerful genetic tool for cutting and splicing DNA. And so this is where, more and more methods have been developed since those times. And it's continuing to reveal more and more, and it's being refined more and more, showing itself as the true revolution in gene editing that it really is. And so what we're talking about is going in and potentially cutting and disrupting any sequence of DNA, and therefore determining its functionality by gene disruption, or in instances, and I won't be able to describe it in this video, but in a subsequent video, how those those cut regions could be edited with target DNA. And so it's basically cut and pasting, very simply, very cheaply. And it began sort of esoterically with the discovery of an adaptive immune system in bacteria. And so Cas9 continues to gain traction, and in recent years, because of its ability to bind to any complementary sequence with the single guide RNA. And so this is a 3D printout, how about that, of the Cas9 protein. Here's the DNA that it's attempting to cut, and here's the single guide RNA annealing to the DNA as well as the Cas9 protein. Pretty cool. Predictably, research papers are increasing year after year after year, and it's exploded after the year 2012. It's making headlines in popular science journals like Science. 2015, the breakthrough year, saying that it's matured into a molecular marvel. That sounds like hyperbole, but I would almost say it's an understatement. And many other important sciences have been jumping on, and there's many important researchers working in the field of CRISPR. Wanted to point out kind of a watershed moment in 2018 where you may have heard of this. It made the news. A Chinese scientist was working with human embryos with the attempt to genetically modify these twins to prevent infection of HIV. And though I don't want to make a statement pro or con on this, it certainly was controversial, and this researcher was highly criticized because, though the attempt is a noble one, a medical attempt to help these girls. When you start to edit the germline as opposed to somatic cells, you're changing sequences potentially that will unfold themselves into the future, and would be difficult to remove once they entered into the human population. And so it's certainly a cause for pause. That when sometimes in molecular biology in particular, things move along very quickly, and sometimes there's ethical considerations whether or not we should do something. Um, People like to talk about designer babies and altering human genome in the future, but in the moment, what we're talking about is curing diseases, and in the moment we're talking about eradicating important problems such as sickle cell anemia and Duchenne muscular dystrophy, just to name a few human ones, as well as altering sequences in agriculture for improved traits. And we're talking about perhaps eliminating bacterial resistance to for disease. So there's a lot of potential good things. However, it should be always considered whether or not human trials should move forward. It should also always be done cautiously. And so in 2019, the first human trials were being put to the test, and more about that in a future video. But I also wanted to finish this historical look at CRISPR-Cas9 by mentioning the fact that Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize in Chemistry in 2020 for their discovery of CRISPR-Cas9 gene editing system. Certainly more to come, substantial in the world of community. And I also wanted to finish by saying, in the decades since the discovery of the restriction enzymes, which have done so much in terms of our ability to genetically modified organisms and cloning, CRISPR-Cas9 researchers are discovering many new molecular tools and techniques that will definitely expand our engineering capacities and capabilities. The question, though, always is it done with the proper ethical considerations, I'll say. And usually the first step of that is understanding. And so education to the concept in general is important. And so that was the impetus for making this video. And so hopefully you enjoyed this brief look at the history of CRISPR-Cas9, and thank you for watching.